to me.
Okay, fine.
Ma'am.
Good evening, everyone. A warm welcome to yet another masterclass series in embryology. Today, we have an esteemed speaker and mentor in the field of embryology. I'll take a few minutes to introduce our speaker, though he is well known among all of us in the field of reproductive medicine and embryology. We've been doing this masterclass series and we take immense pride in introducing SCALE Fellowship Program. It is a one-year fellowship program in clinical embryology and advanced ART. We have 2 seats in each field, and we are very happy to collaborate with IIT Hyderabad for this one-year collaboration for the embryology students, because they will have hands-on on gametes, so that they can have a holistic fellowship program and good mentorship throughout 1 year. And also clinical fellowship will happen at Srichakrafertility, and we also have good hands-on and good clinical case scenarios and good academic programs for the entire embryology and ART program. So we are starting the batch on 25th of July, which is the World IVF Day. So request all of you to pass on this information and utilize this wonderful opportunity. Also, I would like to announce that the next masterclass series will be a continuation of Dr. Sachin Kulkarni sirs. This is on clinical topic, advanced ART. This will be on 24th of July at 7:30 p.m. This is on novel stimulation protocols. This is part 2. We already had the part 1 session earlier. And on the embryology masterclass will be on 7th of August. That is on creating life in a dish, the critical role of embryo culture media. This will be delivered by none other than Dr. Sanjay Shukla. I take immense pleasure in inviting Dr. Vijay Mongoli, sir. He's a lab director at Fertility Clinic, Mumbai. He's associated with human IVF since 1986. We all are benefiting from all the work all these mentors have done today. We have all the ready-made recipes for us to really, work on the patients. They have worked for decades to give us this platform. And he's the Secretary General of ISAR, Chairperson of Embryology ISAR. He's got very large number of publications and in peer-reviewed journals. And he's got presented, he's a well-known national and international figure, a lot of chapters. He has contributed in medical reference textbooks. He's a recipient of Lifetime Achievement Award in 2017. And he holds a patent for an indigenous vitrification device can you just take it up? Little bit up.
Maximum. That's the maximum.
I'm not able to see.
Vitrification device for the cryopreservation of gametes and embryos.
Yeah. We welcome you, sir. Today, sir is going to take us through the sperm preparation techniques, the entire science behind it. All of us do it routinely in our lab. Is it that simple, or we need to take a lot of precautions on how to prepare sperms for different case scenarios, whether it is conventional IVF, ICSI, or preparing the sperms for microfluidics or MAX or anything? We are going to, or the testicular sperms. So we welcome you, sir, and please mentor us in the next 1 hour so that we'll take home a lot of practical points and learning from your talk. Welcome you, sir. Thank you.
Sure. Okay. Fine. Okay. Can you see it now? Can you see it?
Yes, sir.
Yes, sir.
Okay. Fine. So thank you, Dr. Vasundara. Thank you, Dr. Sindhuja, and the complete team of Srichakrafertility for inviting me to such an interactive masterclass on ART involving embryologists, the clinicians, and PG students. Precision in motion, exploring the science of sperm preparation. Well, it is truly unfair on the part of nature that in the whole process of reproduction, all a man has to do is contribute a single sperm, and in most cases with pleasure. And a woman must take the responsibility for all the remaining stages, right from release of an egg to delivery. Now, though a single sperm is required to fertilize an oocyte, in a reproductively healthy man, millions of sperm are produced in the testes. I use the word reproductively healthy person because reproductive ability need not be associated necessarily with physical characteristics. A he-man may not have a single sperm in his ejaculate, whereas a person weighing hardly 30 kilograms may have more than 200 million sperm per ml. A sperm is a very fascinating yet mysterious entity. Before discussing the separation techniques, let's understand the basics of spermatogenesis, which is necessary to comprehend why sometimes even ICSI doesn't result in fertilization and why occasionally the fertilized oocyte displays multiple pronuclei. In males, the process of meiosis, that is sperm formation, it begins at puberty and continues throughout the life. And one diploid spermatogonium with two sets of chromosomes, one from each parent, after two divisions, it produces four haploid sperm, each with a single set of chromosome having genes exchanged from both the parents. A very straightforward phenomena. Unlike in females, where the oogenesis starts when the fetus is still in the mother's womb, here the meiosis begins, but the process gets arrested at a particular stage. It resumes in puberty, but again gets arrested, waiting for the sperm to come. And after all this, one cycle of meiosis results in a single fertilizable oocyte before terminating the process of egg formation at menopause. See, even at this early stage, women are so complicated and difficult to understand. Coming to men, a diploid germ cell, that is a spermatogonium, has a typical nuclear status of two chromosomes, two chromatids. One chromosome from mother and second from father. Now here small n denotes the number of chromosomes and capital N represents the number of chromatids. We know that for fertilization, we need two haploid cells, one from male and one from female, having a nuclear status of one chromosome, one chromatid. Now, it would have been a straightforward step for the nature to divide this spermatogonium into two parts. Just divide cytoplasmically and nuclear-wise, creating two haploid cells. So each one of them can unite with one oogonium and fertilize it. But it doesn't happen that way, and there is a reason for it. As a first step, the cell undergoes DNA replication, thus acquiring two additional sets of chromatids and becoming a two chromosome, four chromatids structure. Now keep in mind that the number of chromosomes remains the same, that is two. Only the chromatids double, which are attached to the centromere. It then undergoes a recombination or crossover stage, which is primarily responsible for exchanging the phenotype traits and other genetic characteristics from both the parents. This is the purpose of DNA replication. Now that it has two chromosomes and four chromatids, it must obviously undergo division two times to attain a haploid status. And that's what happens in the form of meiosis 1 and meiosis 2. Meiosis 1 is nothing but first cell division, and meiosis 2 is the second cell division. So during meiosis 1, both sets of crossover chromosomes, they are pulled apart through the spindle. Meiosis 1 occurs, resulting in the formation of two cells, each with equal cytoplasmic and nuclear division, having a nuclear composition of one chromosome, two chromatids in each cell. Now, theoretically, this cell has a haploid status of one chromosome, but it still has two chromatids. So as we need one chromosome, one chromatid structure, therefore, it must undergo one more division to reduce the number of chromatids to one. So it undergoes meiosis 2, which ultimately yields four haploid spermatids, each with a haploid status of one chromosome, one chromatid, having genetic material exchange from both the parents, thus acquiring the necessary nuclear maturity. Now remember, this is still a nuclear maturity. Now to achieve the cytoplasmic maturity, these cells undergo spermiogenesis after entering into the epididymis, completing their cytoplasmic maturation process and producing four morphologically and chromosomally compatible sperm. Now, if the whole spermatogenesis is so standardized, then what can go wrong? Sometimes, particularly during meiosis 2, the nuclear division can be unequal. Both the chromatids may migrate to a single cell, and that is one of the reasons for either no fertilization or multiple pronuclei. So these sperm thus form They are then ejaculated through the semen and are ready to fertilize the oocyte. So to assess a man's overall fertility potential, we perform a semen analysis, which is the simplest and the quickest way to evaluate his reproductive ability. However, we should keep in mind that semen analysis is not a test of fertility. It is an indicator of the functional status of the male reproductive tract. Therefore, whenever we get two successive abnormal semen analysis reports, a complete evaluation by an andrologist should be performed. Now, regarding semen evaluation, the most crucial fact to keep in mind is there is a drastic difference between the semen analysis performed by a pathology laboratory and that done by an embryologist. Both have different perspectives to evaluate a male's reproductive ability. The pathology laboratory uses WHO standard values to compare and interpret whether a semen sample fulfills the criteria of a fertile man. However, the fundamental threshold values and cutoff points for the natural conception differ significantly from those for assisted reproductive technology procedures. WHO criteria are meant for natural conception, and we are talking about ART procedures. Therefore, when we discuss the ART procedures, we need to think beyond the WHO manual. Because in ART, we can select a population of the sperm and can even manipulate them to some extent. And therefore, the cutoff values for both are different. For example, according to WHO manual, the minimum requirement for a man to father a child is the sperm count of 15 million per ml, motility of 40% with 32% progressive motility. However, in ART, a 5 million initial count with a post-harvest count of 2 to 3 million, greater than 90% motility, and a grade of 2 to 3 can certainly be considered for at least 2 IUI cycles. Now, these cutoff values further decrease for IVF and for ICSI. For IVF, whatever may be the initial count, if we can get a minimum of 1 million harvest count with 3 to 4 grade, we can still use it effectively for insemination. And for ICSI, as we all know, we just need one viable morphologically normal sperm per oocyte. Therefore, it is very crucial and very necessary that an embryologist should perform at least one semen evaluation before starting the treatment. For ART procedures, we have different yardsticks and categories to consider for male factor indication, which are not covered or even mentioned in the WHO manual. Male factor can be divided into 3 main categories. Where there is a normal spermatogenesis, the sperm are found in the ejaculate, but the patient is still considered as subfertile. This includes oligoathenoteratospermia, globospermia, those with immotile cilia syndrome, and those with sexual dysfunction. Second category is where there is normal spermatogenesis, but the ejaculate does not contain the sperm. That includes obstructive azoospermia and retrograde ejaculation. And the third category includes those patients with impaired spermatogenesis, like Sertoli-Cell-Only syndrome or maturation arrest. Now let's see how to retrieve sperm from these various categories. Oligoathenoteratospermia. Now, although by definition, these are 3 distinct categories: low count, reduced motility, and high abnormality. But quite often, it is observed that they are found together and hence clubbed in a single category. Therefore, a standard protocol can be applied to obtain healthy, morphologically normal, and usable sperm from this category for fertilization. But depending upon the extent of oligozoospermia, if the count is between 5 and 15 million per ml, with a grade of motility in the range of, say, 2 to 3, and the semen volume is adequate, then the first practical way to obtain maximum sperm by natural means is to ask the patient to collect the sample in split ejaculation. Believe me, you will get surprisingly high concentration in the first split. Secondly, dividing the raw sample into 2 aliquots, followed by the swim technique, may yield sufficient sperm for IUI, even for the conventional IVF, and definitely for ICSI. This is how the sperm Sperm is a simple method based on the natural tendency of live sperms to migrate from denser pellet to the above layered liquid medium for their free movement. It is generally used for normosospermic patients with less debris. Patient is asked to collect sample, preferably by split ejaculation. Collection bottles are clearly marked with patient's identification. A 15 ml tube is marked with patient's name. Liquefied sample is taken in a sterile test tube. A drop of it is evaluated for sperm concentrations, motility, and presence of debris, dead sperms, leukocytes, or cells. Sample is mixed with about equal volume of sperm wash medium. Another tube with equal volume is taken as a balance tube. The tube is centrifuged at 200 gyrations for 5 minutes. We should keep in mind, gyration is not equal to RPM. We have to convert using this formula, as RPM can vary for different centrifuges. After 5 minutes, tube is taken out slowly and supernatant is removed carefully without disturbing the pellet. A distinct pellet should be clearly visible. The pellet is layered with 1 to 1.5 ml medium. And kept at an angle in a beaker with a loose cap to enable pH maintenance. The beaker is kept in an incubator for up to 30 minutes, again depending upon initial count and grade of motility. Depending upon sperm count and percent motility, supernatant can be removed between 5 to 30 minutes post incubation. The harvested outcome is evaluated. It should have 100% motile sperms with improved grade of motility without debris. So this was regarding the SIMOP technique, how it can be done for the almost normosospermic sample. Now, if the sample has 5 to 10 million sperm count contains more debris, dead sperms, such as leukocytes and pus cells. Then a density gradient is a better option, yielding a clear motile sperm fraction. This is how the density gradient is done.
Gradient technique. Gradient method is based on separation of cells according to their differential densities. Silane-coated silica particles form smooth gradients on centrifugation and trap cells of different sedimentation coefficient. This method is particularly useful for asthenoteratospermic samples having less motility and high percentage of abnormal spermatozoa. Gradient technique effectively concentrates morphologically normal spermatozoa. Patient is asked to collect sample, preferably by split ejaculation. Collection bottles are clearly marked with patient's identification. After proper liquefaction, a drop of sample is evaluated for sperm concentration, motility, and presence of debris, dead sperms, leukocytes, and pus cells. Gradients of 90% and 45% are used, which are readily available. One ml of 45% gradient is overlaid on 90% gradient carefully without mixing. Liquefied semen sample is layered on 45% gradient. Sample tube along with balance tube is centrifuged at 300 gyrations for 20 minutes. After centrifugation, tube is gently taken out. Cells of different sedimentation coefficient get trapped at different levels. Upper part is semi-plasma without any cells. Leukocytes and pus cells remain above 45% layer. Dead sperms and debris are trapped between 45% and 90% layers. And morphological normal sperms form the pellet at the bottom. Pellet is carefully removed and resuspended in 0.5 to 1 ml medium. A drop of resuspension is evaluated for sperm count and morphology. It should result in higher concentration of morphologically normal sperms with improved grade of motility without debris. These sperms can then be used for either IVF or ICSI.
So friends, these were two most commonly used semen preparation techniques to obtain, morphologically normal sperm as far as possible, wherever. And globally, they are the standard procedures. So I had shown them a little bit in more detail. Now coming to other parts, other retrieval techniques to obtain the sperms. Intracytoplasmic morphologically selected sperm injection Now, in severe teratozoospermia, we can select morphologically normal sperm with higher magnification, or which is called as IMSI. Now, though very little is mentioned in the WHO manual about the vacuoles as such, the presence of vacuoles, particularly towards the anterior side of the sperm head, is known to be detrimental and has an adverse effect on the fertilization, subsequent embryo development, and also it results in increased abortions. So using optical magnification of 600 times or digital magnifications of 6,000 times, it is possible to visualize the size, type, and position of the vacuoles in the sperm head and select the one without vacuole. Now, also, although at present the concept of IMSI has almost become obsolete, and there are many papers published that it really doesn't have that much of a significance, and even the donor sperms contains more than 50 to 60% vacuoles. So there is a doubt whether the presence of vacuoles has that much of a significance. Coming to physiological intracytoplasmic sperm injection, PICSI. This mimics the natural oocyte interaction by selecting the sperm that binds to the hyaluronic acid. Significance? These sperm known to show better ecology, DNA integrity, and maturity. Now, they are especially relevant in better sperm selection for ICSI. Globozoospermia. Now, this is still a mystery and a poorly understood category because theoretically these sperm don't have an acrosome, which is not required anyway when we do the ICSI. But still the fertilization rate is poor in such cases. Now, such round-headed sperm, they have poor fertilization because they lack the surface proteins that activates the oocyte that initiates meiosis II. So the oocyte doesn't acquire the mandatory haploid status and thus fail to initiate the fertilization in spite of a sperm entering into the ooplasm. Now, there is no technique available that can distinguish between the round-headed sperm and the normal oval sperms. This must be done manually by searching extensively within the cluster of the sperms. Now, it may be possible to get fertilization with round-headed sperm after oocyte activation in some cases. We performed oocyte activation with calcium ionophore in 3 instances of globospermia, with 2 resulting in pregnancy. Immotile celiac syndrome. When we observe such sperms without motility, then we have to first confirm whether they are non-motile or non-viable, keeping their viability intact in an easy manner. This can be checked by 2 methods, by HOS test and by using pentoxifile. Now, we can also check it by staining method, but however, as we all know, obviously we cannot use them for actual fertilization purpose. Obstructive azoospermia. Now, this is relatively easy to tackle for both the andrologist and for the embryologist, because generally enough sperm are obtained from the epididymis because the sperm formation is already there. It is just that because of some obstruction, they are unable to come into the semen. The procedure can be done on a trial basis, and the sperm obtained can be frozen for later use. This is how it is done. The sperm can be retrieved from the epididymis. The needle is inserted at the flabbiest part of the epididymis that has the maximum probability of obtaining the sperm. The fluid is aspirated and then it is handed over to the embryologist. And this is how the sperm appear under the microscope. Usually sufficient sperm are obtained that can be used for IVF or even sometimes for IUI if they have more than enough motility, grade of motility. Retrograde ejaculation. Now, it is a condition where the ejaculated semen enters the bladder instead of oozing out through the penis, mainly due to the issue with the bladder neck muscle. Now, this bladder neck muscle, it contracts in such a way that the semen enters into the bladder. Now, generally, a dry orgasm is the primary symptom of the retrograde ejaculation. Now, in such a situation, usable sperm can be obtained by making the urine alkaline over a prolonged period of about a week. And on the day of treatment, ask the patient to masturbate, collect the alkaline urine, and process it immediately. Most of the time, sufficient sperm are obtained that can be used even for IUI. Non-obstructive azoospermia. This is the most difficult male factor category to handle. A lot of uncertainty is here because of highly focused spermatogenesis. Out of the millions of the tubules, only certain parts of the tubule they produce the spermatozoa, and that's why it becomes extremely difficult to find out that particular part where the sperm's formation is taking place. We had stopped doing the trial TCA for the same reason, because even if you get the sperm during the trial, there is no assurance of getting them at the time of ovum pickup, and vice versa. Even if you don't get the sperm at the trial, that doesn't mean that you won't get it at the time of ovum pickup. There are some publications on preserving the sperms obtained during the trial TCA, but their efficacy it varies. Using intermittent stages of the spermatogenesis, such as spermatocytes or spermatids, is also not a compelling and convincing approach. Sometimes the only option left for such a couple is that of a donor. This is how the TCA is performed. The seminiferous tubules are extracted from the testes. A butterfly needle is inserted through the testes. The tubules are trapped from various positions into the needle. By doing it upside down, up and down, and trying to get from different positions. Slowly, the needle is taken out. After the embryologist is convinced that sufficient tubules are already trapped, the trapped tubules are extracted using the forceps, which are passed on to the culture room. This is how the maximum tubules are collected. Then initially they are collected in the buffered culture medium, and then they are passed on to the culture room. This is the butterfly needle is flushed. The tubules are taken in a glass dish and teased in such a way so that the inner content it completely gets oozed out into the surrounding medium, and then that surrounding medium is extensively hunted for the presence of the sperm. This is how the TCA sperm material, the extracted material from the tubule appear under the inverted microscope. Most of the time, the sperm, if obtained, they may be non-motile because they have not yet completed their cytoplasmic maturity. But occasionally, you may get the sperm with twitching motility. Again, here, incubation of these sperm for 2 to 3 or using petoxyphallin or theophylline may induce motility and can be successfully used for insemination through ICSI. Microfluidics. Well, microfluidics is an emerging technique in assisted reproductive technology that offers a more physiological, efficient, and gentle method of sperm preparation for in vitro fertilization. Traditional methods like density gradient centrifugation or swim up, they may expose the sperms to the oxidative stress or mechanical damage. In contrast, microfluidic devices are microchannels. They use microchannels to mimic the natural selection process of the sperm migration, allowing only the most motile and morphologically normal sperm to be selected. Now, these systems separate sperm based on motility and morphology without the need of a centrifugation, as I mentioned, and thereby reducing the DNA fragmentation and preserving the sperm integrity because they are not exposed to any mechanical damage. Microfluidics can enhance the fertilization outcomes, especially in cases of male factor infertility. And moreover, the method is time efficient. It requires minimal handling and can be easily standardized, making it a promising tool for improving IVF success rates and embryo quality. These are various types of devices available to separate the sperm based on the principle of microfluidics. This is Zymote, this is Lensuk, and this is Qualis sperm sorter. Coming to the advancements in sperm separations. Nanotechnology in sperm separation. Nanotechnology is emerging as a promising frontier in assisted reproductive technologies, particularly in the precise and efficient selection of the sperm for IVF. Traditional sperm preparation methods like swim up and density gradient, they are limited by their ability to eliminate all forms of DNA damage or apoptotic sperms. Nanotechnology offers novel tools that can enhance the selection process with high specificity and sensitivity. Magnetic sperm sorting. Magnetic sperm sorting, also known as magnetic activated cell sorting, MAX, is an advanced technique used in ART to select the healthier, more viable sperm for IVF procedures. This method utilizes magnetic nanoparticles coated with annexin 5. This is a protein that binds to phosphatidylserine, which is a marker present on the membranes of the apoptotic or dying sperm cells. So during this process, the semen is passed through a magnetic column. The sperm cells with the markers of the apoptosis, they bind to the magnetic particles and they are retained on the walls. While the non-apoptotic healthier sperm, they pass through and they are collected and used for the fertilization. Advantages, well, it is reported that it improves the sperm quality by eliminating apoptotic and DNA damaged sperm, potentially increases fertilization rates and embryo quality, and may reduce miscarriage rates in selected cases. MAX is often used in conjunction with other sperm preparation techniques like density gradient centrifugation, particularly in cases of male factor infertility, repeated IVF failure, or recurrent miscarriages. This is because one cannot pass raw sample directly through this column. But then again, it raises the question that if already some part of centrifugation is done, then how effective that may be in getting maximum motile sperms. But this is to select the remaining motile sperms after the either swim up or density gradient techniques. Researchers are also working on other possibilities involving nanotechnologies. For example, nanostructured surfaces. They create artificial surfaces. The surfaces are engineered at the nanoscale that can mimic the natural environment of the female reproductive tract, allowing the selective adhesions and motility-based sorting of the high-quality sperms. Now, this biomimetic approach favors the selection of morphologically and functionally superior sperm. DNA integrity sensing. Well, this is also interesting. Nanosensors are being developed to assess the sperm DNA integrity in real time. Now, these devices can help in discarding the sperm with DNA fragmentation or chromatin abnormalities, which are not always detectable by routine microscopy. Now, such type of non-invasive technique is the need of the time, because whatever techniques currently available to detect the DNA fragmentation in sperm, they use stains and thus making those sperm non-usable. Targeted drug nutrient delivery. This is also a new invention type of things which is taking place, which is not yet applied routinely. Here, the nanocarriers can be used to deliver the antioxidants or energy substances or substrates to the spermatozoa during preparation. That means while you process the semen samples by using either swim technique or density gradient technique, these chemicals are added into that, improving motility and viability without causing oxidative stress. However, we should keep in mind that these advancements are still in the experimental stage and have not yet been implemented in routine use. Coming to the omics in sperm preparation for the IVF. Well, the application of omics technologies, including genomics, transcriptomics, proteomics, and metabolomics, is revolutionizing the field of sperm selection, particularly in IVF. So these advanced approaches, they provide deep molecular insights that extend beyond the conventional parameters such as morphology and motility, enabling more precise identification and selection of functionally competent sperm. Genomics. Well, genomic analysis, it identifies genetic mutations, chromosomal anomalies, or structural DNA abnormalities in sperm. Techniques like comparative genome hybridization, CGH, and next generation sequencing, NGS, it can detect the sperm with Y chromosome microdeletions, aneuploidy, DNA fragmentations. Now, here the sperm with intact and normal genomic profiles can thus be preferentially selected to improve the fertilization and embryo quality. Transcriptomics. Although mature sperm, they are transcriptionally silent. Residual RNAs, that content reflects the sperm's developmental history and functional potential. By analyzing the sperm RNAs like mRNA and microRNAs, transcriptomic profile, it can distinguish between the fertile and subfertile sperm populations, identify markers for motility, capacitation, and fertilization capacity. It aids in non-invasive sperm quality assessment. Proteomics. Well, proteomics focuses on identifying and quantifying the proteins involved in sperm function. Mass spectrometry-based techniques, it can detect the surface proteins or intracellular markers linked to the sperm vitality. It differentiates sperm based on the capacitation, status, or acrosomal integrity. It reveals the proteomic biomarkers associated with fertilization success or failure. Metabolomics. Well, this field evaluates metabolic signatures like lactate, ATP, ROS that reflect the sperm functionality. Using techniques like MMR spectroscopy or LCMS, metabolomics can assess mitochondrial activity and energy production, evaluate oxidative stress and lipid peroxidation levels, identify sperm populations with optimal metabolic fitness. Well, omics has some advantages, like it enables precision selection of sperm with the highest fertilization potential. They offer non-morphological markers of sperm health. It supports personalized IVF strategies. It helps in identifying male factor infertility causes at the molecular level and can improve the IVF outcome and reduce miscarriage rate. But it also has some challenges that restricts its use routinely, like high cost and technical complexity, requirement of specialized equipments and expertise, and lack of standardized clinical protocols for routine use. Coming to the last aspect, artificial intelligence in sperm preparation. Well, artificial intelligence is increasingly being integrated into the sperm preparation process in IVF to enhance the accuracy, efficiency, and outcomes. Traditional sperm selection, it relies on manual observation of motility and morphology, which can be subjective and prone to inconsistency. Whereas AI introduces automation and objectivity by analyzing the large data sets using machine learning algorithms. Now, AI-powered systems, they can assess the sperm motility and morphology with high precision using image recognition, identify and rank the healthiest sperm based on multiple parameters like DNA integrity, movement patterns, and shape, and predict the fertilization potential by learning from the previous IVF cycle outcomes. Advanced platforms also combine AI with microfluidics or robotics to automate the sperm sorting and selection, reducing human error and variability. This technology holds promise in improving the success rates in IVF, especially in case of male factor infertility and repeat IVF failures. However, we should keep in mind that the capability of AI to predict the outcome, it entirely depends on the data fed to the computing machine, which includes various observations from different centers. Therefore, it becomes imperative that all the observations be carried out according to a standard protocol and uniformly by all the investigators. Otherwise, the basic purpose may be compromised. So friends, these were some of the points related to the recovery of the eligible sperm for fertilization. Sperm preparation is not merely a mechanical separation. It is a precision-driven, biologically informed process that bridges the natural selection and assisted fertilization. Advancements in the sperm selection aim to improve the ART outcome by focusing on genetic integrity, biochemical functionality, and reduced cellular stress. Now, as technology evolves, sperm preparation continues to refine the very essence of precision in motion, selecting the right sperm at the right time for optimal reproductive success. Thank you very much.
Thank you so much, sir, for taking us through physiology of spermatogenesis, the basics of swim up. I think proper swim up now is, the need of the hour, and you took us through a video demonstration of swim up and density gradient, took us through all the sperm preparation techniques, gave us a lot of practical points. I think now Sindhu can, start taking the poll, and we can simultaneously take the Q&A, sir, with your permission.
Sure, sure.
We have a couple of questions in the chat box, and Sindhu, please kindly take over.
Yes. So I've shared the questions. All of you can attempt. Meanwhile, we'll ask Sir some questions. So thank you so much, sir. That was a very elaborate presentation. You didn't even take a break for 5 seconds in the past 45 minutes.
Thank you.
So the first question on the chat box, sir, is what is split ejaculation?
Well, split ejaculation means asking the patient to collect the sample in two parts. It is not 2 times collection, remember. Because many times there is a misconception that ask the patient to give the sample 2 times. No. A single sample, single ejaculate has to be divided into 2 parts. See, physiologically, when the ejaculation takes place at that time, the very first fraction that is ejaculated is full of rich of sperm fraction and the prostate glands. So if the patient can differentiate between first 2 or 3 forceful parts from the rest of the semen sample that contains only seminal plasma and the coagulating factors, then naturally we may be able to get a sperm rich fraction in the first split. So we need to give patient 2 bottles. Of course, let patient get accustomed to this kind of collection, because generally all over the world, almost all the people, they give a single bottle and the patient collects in the single bottle itself. So it becomes slightly difficult for the patient. So proper instructions are to be given to the patient how to collect it. If possible, he can take help of his wife, but not to waste particularly the first fraction of the semen, which should not fall on the floor. And we also should instruct him that in case if it falls down, please do not pick it up and put it back into the bottle. It is very, very, very important, because we have the experience of this. Patient will not tell you that this has happened, and you will get a complete heavy contamination while processing the sample. So split ejaculation is a very, very effective way of separating naturally the sperm rich fraction.
Very interesting, sir. I don't think many people follow this. So do your patients find it convenient to follow it now?
Yes, yes. See, once you give proper instruction to them, ask them how to hold both the bottles in one hand, or as I mentioned it to you,
yes, sir,
without feeling any shame, they can take help of their wife. See, ART is a treatment, and our aim is to give them their baby. There is no need to feel ashamed that, oh my God, my wife is coming with what the other people will think. Nothing like that. So it is important that we should counsel the patient properly, that it is important for the laboratory to get sample properly. If this is counseled properly, then definitely they will understand it, and even they will collect it properly.
I think, sir, most of us do keep a backup semen sample freezing, right? So on the day of, egg retrieval, we don't have to stress them with this. Maybe at the semen freezing itself, if we, kind of tune them, it will be...
Exactly. Yeah. That is what I was coming to, because it is a must that we should keep a backup sample for enormous reasons. I have seen n number of cases where very, eligible patients who don't have any kind of reservations of giving the sample throughout their life, but on the day of ovum pickup, particularly if less number of eggs are found, or thinking that someone from their family is sitting outside, they just get nervous and they are unable to give the sample. So occasionally it may happen that the business person or the service people like from Navy and fields, they may have to go on the field and they are not there at the time of home pickup. So these are the situations where you have to keep a sample backup, and at that time only if you give them such instructions, then they are used to it.
Thank you, sir. Sir, and sir, in the videos that you showed, sir, so for simple wash, you prefer round bottom tubes, and for density gradient, do you prefer conical centrifuge tubes?
Nothing like that. It is not like
Okay.
that. It
Is it
entirely
any
depends
different?
upon the sperm, because see, as I mentioned, generally normospermic sample we use swim up, so there are sufficient sperms available. If the number of sperms are less, then it is better to use a conical one so that they will concentrate at the bottom. But otherwise, you get a nice flat pellet if you use round bottom these things, and the surface area is more, so more number of sperms can migrate upwards.
Great. Sir, and you had also shown very comprehensive videos of how a TISA sample looks, how a PISA sample looks. So for these kind of samples, sir, do you prefer simple wash or density gradient? And do you use erythrocyte lysis buffer for these kind of
Yes,
samples?
erythrocyte dyes buffer can be used if, in case, if the entomologist hands over you the tissue or the culture medium with or the fluids with filled with blood, you can use it so that it becomes slightly easier for you to screen it. As far as the use of other either swim or the density gradient is concerned, density gradient I don't think will be a viable option. We have used a single gradient method in some cases where a moderate debris is found along with the sperms, because see, the more are the columns you create in between the that not semen as such but the secretions and the bottom part, more are the chances that you may lose the viable sperms in between the columns. So a single column of 90% will also do, and that also helps in certain cases. Yes, if the count is very good, then you can go with the swim hole also. It also will give you sufficient sperms.
And if you're using pentoxiflin or any motility stimulating agents, at what point of time do you prefer adding them, sir? After the simple wash to the pellet? I'm asking for the benefit of all the students so that they're aware of how to do it.
Yes. Now it has two parts in it. See, if initially the count is adequate, but the percent motility is very less, in say around only 10% are motile or 15% are motile, then to make the sperm motile first, you can add the pentoxifylline before you centrifuge,
Okay.
and then you can then make more number of sperms, they may acquire the motility and you can get them. But if the majority of the sperm are non-motile, like in TC sperms, then you have to add it just before you go for the ICSI. You cannot add the such pentoxifylline or theophylline for a longer period of time, because the ATP generation is it works like a rhythmic manner, and it immediately it goes up. The sperm acquire their motility within around 5 to 7 minutes, and then they go to the peak. It depends upon that particular sample and the secretions into that, but then it reduces also abruptly. So after say around 15-20 minutes or half an hour, you may not be able to get any motile sperm from that. So it is a very short duration that you have to expose these kinds of chemicals.
So very,
So
very
for
well.
checking the motility in the same vein, have you tried laser shots to check motility for the sperm? Can we do it in a controlled manner? Is it difficult?
You can do it, but it is, see, you can do one at a time. It takes a longer time. You need a precise firing for that. Instead of that, this is much easier, because in a one droplet, a bunch of the sperm, they can be, you can visualize them, and you can immediately get an idea that what is the proportion of the sperm are viable and non-viable.
So there are some more questions on the chat. So what do you prefer, double density or triple density? And for density gradient, what is the preferred time and RPM that you use?
Well, as I mentioned, the double density is generally used and generally sufficient, because double density almost filters all the debris part of it, and you really get a good sperm, as I showed it to you. What is the next question?
What is the time and RPM that you prefer for density?
RPM, as I mentioned it to you, 300. This requires slightly more the gyration. I calculate in terms of gyrations because, as I mentioned it to you during the video,
Yes, sir.
you calculate your RPM as per as the gyration is concerned, because the length of the tube and the length, the distance between the center of the centrifuge and end of the tube, that makes a lot of difference. And this you calculate first, that is the radius, and depending upon that you calculate how much should be the RPM. Now that RPM varies for 200 gyrations and 300 gyrations. So 300 gyrations is standard for the density gradients, and 200 gyrations is for the swim technique.
Yes, sir. So some have asked, after density gradient, do you allow to swim up for 20 minutes? I think, like you said, it depends on the type of sample again.
Type of sample. If in spite of after density gradient, if you are getting silt debris into that one, then you can go a mild swim up technique. But just remember, again you are giving centrifugation to that. So again you are increasing the probability of the ROS productions.
So next question is, in which sample will you not advise microfluidics?
For the TISA sample where you don't see the motility at all. Microfluidics, based on, they expect that the device manufacturers, they expect the sperm to travel from one part of wherever you put that droplet, to the other end. So obviously the first basic requirement is that the sperm should be motile. If the motility is less, very less, just twitching motility, they will never be able to migrate and cross that channels and come. So that is one. You can add the pentoxypalline and then ask them to, migrate it. That is possible.
Okay. So this is somebody who has asked a very smart question. So they're saying that microfluidics and swim up has almost a similar mechanism. But when you don't do swim up, then you're avoiding the ROS. So are you saying microfluidics is superior to swim up?
Microfluidics is superior because in swim up you are centrifugation. Remember one thing. This is a basic difference between the microfluidic and the swim up is this centrifugation. So if the sperms are already weak, oligospermic and astenospermic, and again you are giving them your 200 type gyration centrifugation, you are creating a little more damage than what is in the natural status. In microfluidics, you are not doing anything to them. You are just depending upon their natural tendency to swim up.
Yes. Thank you, sir. Ma'am, it's been
I-
11 minutes. Shall
Yeah,
I?
you can take the poll. There are a couple of more questions. Probably you just take the first 5 questions and then I'll give Sir some voice break before he answers a couple of more questions.
No problem. You can just go ahead. No problem.
Okay. I've just ended the poll and I'm sharing the results. Okay. So the first question was, in males, spermatogenesis, 69% answered correctly, begins at puberty and continues throughout life.
This is important and this is interesting because, as I slightly mentioned, because the oogenesis I did not cover in detail because that is out of the scope of this talk. But it is equally interesting to know, how the oogenesis takes place and what is the difference between spermatogenesis and oogenesis. So it begins at puberty and continues throughout the life, whereas in oogenesis there are stages.
Correct. Yes.
And it starts in utero. It gets arrested at one stage. It again starts during puberty. It again starts at second stage. Again, it starts waiting for the sperm. So it's a little bit complicated. And it ends at menopause. As against the sperm preparation, it continues throughout the life.
Right. Next question was, one meiosis cycle of spermatogenesis produces dash mature spermatozoa. The correct answer is 4 and 70% have answered it correctly. The next question is, semen analysis is a test of fertility. So sir very well mentioned in his presentation that it is not a test of fertility. Rather, it is a test of the function of the reproductive tract and the system. So the answer is false. The correct answer is false. The next question is, natural conception and ART procedures have the same cutoff values. A majority have answered false, which is correct. The main concern of centrifugation in swim up and density gradients is 55% have answered it correctly, increased ROS production. Immotile cilia syndrome has only non-viable sperms. False, correct. That is, there will be viable sperms which can be identified by addition of motility stimulating agents. IMSI detects acrosome content of sperm. It does not detect the acrosome content of sperm. The correct answer is false because we can only assess the morphology, the presence of vacuoles, and overall morphology of the sperm. So the content of the acrosome cannot be identified. So I think, sir, that was a very tricky question and majority fell into the trap. And Pixie dish uses hyaluronic acid. That is 63% have answered correctly. Microfluidics is primarily used to obtain sperms with good motility and DNA integrity. 95% have answered correctly. In MAX, the nanoparticles are coated with annexin V, that is annexin 5. 53% have answered correctly. Sperm RNA, example, messenger and microRNAs, can distinguish between fertile and subfertile sperm population. 86% have answered true, which is correct. Proteomics focuses on, the correct answer is all of the above, which is identifying and quantifying proteins, detecting surface proteins or intracellular markers, and differentiate sperm based on capacitation status or acrosomal integrity. All of the above, 60% have answered correctly. Efficiency of AI in sperm preparation depends upon all of the above again, which is accurate data feeding, uniform protocol, and meticulous data analysis. So majority have gotten everything correct, which means most of them have paid attention, sir, and the presentation was very clear.
Good, I'm
Thank
happy.
you, sir. Yeah. Sir, with regards to retrograde ejaculation, can you tell us how to prepare the sample for IUI? Like, you mentioned that 1 week of alkalizing agents and, all this.
Yeah,
So,
right.
can you just tell us? Yeah.
Correct. Correct. It is a combination of the efforts by andrologist and the embryologist, because the andrologist should first, take care from the clinical aspects, the making the bladder, alkaline. So he gives the medicine to the patient in a controlled manner, and then after 1 week, at the time of the giving the sample, just ask the patient to masturbate, and immediately after masturbation, as I mentioned it to you, that it is a dry orgasm. He will feel that he has got the orgasm, but there will not be any ejaculation as such. The moment he feels that there is ejaculation within the body, he immediately should collect his urine. And once he collects the urine, depending upon the volume of the urine, you can divide it into 2 or 3 or even up to 4 parts, in the conical tube, or the even round bottom tube as well, and then process it the same way as you process it for the swimmer. So you will be able to get it first centrifugation. You may get the pellet itself, and then you can allow that pellet to undergo the swim technique, and you will get sufficient sperms. See, here, remember one thing, that retrograde ejaculation, there is no functionally any wrong with the patient. The sperm formation is there, the spermatogenesis is taking place properly. It is only the oozing out part, is not correct. So rest, everything remains the same as a person with a normal spermatogenesis or normal semen parameters. So his count, in most of the cases, they are normal. So sufficient sperms are available.
So, in your experience, we know that literature is quite confusing in terms of raised DFI samples. Apart from lifestyle modifications and everything, we have a lot of these sperm selection methods like microfluidics, MAX, Pixie. Is there one that is superior to the other, or microfluidics takes a superiority amongst others because we don't centrifuge the sample? That is
Yes.
one. And second thing is this MAX versus microfluidics. Do you think still microfluidics is better, because again MAX we have to density gradient and then do it?
Right. I am quite for the microfluidics, particularly for as far as the ROS production is concerned. It really helps us because it is the most natural way to get the sperm without allowing them to undergo any kind of physical stress. So I definitely prefer it. What was the first question you asked me?
In raised DFI samples, like which kind of sperm
Yeah, right. So
preparation?
microfluidics is the preferred one. But one thing you remember in the DFI, as I mentioned in the talk, the assessment of the DFI is an invasive method. So all you get is an overall idea about the proportion of the sperm in that particular population about the positive and negative things. Practically, it is of no use to us while selecting the sperm for doing the ICSI. Can you select that particular part? I again and again tell the embryology is the same thing. If the patient will come and talk to you and then you do his DFI, if you find the DFI is very high, then you will tell the patient that, oh, I'm very sorry that you are, say, very high DFI and almost 90% of your sperm, almost 90% of our sperm are abnormal. The patient,
Please mute, excuse me.
the patient will immediately tell you, I don't care about it, doctor. 10% are still normal. So you select from that 10%. So that is what is expected from the patient, from us. Can we do that after doing this? No, it is the DFI evaluation is good enough for the purpose of counseling the patient. If there is less fertilization, then you can tell that why there is less fertilization. That is what is possible, but you cannot select that sperm, the normal sperm, and do it. So the only way is to minimize the DNA fragmentation probability by selecting the most natural way, the way you can select the sperms from the sample.
So if there is a raised DFI and we do 2 ejaculates on the day of egg collection and we do microfluidics, we can still do conventional IVF?
You can definitely do it. It helps.
Sindhu, is there any questions on the chat box? So what is your sperm preparation methods and how much volume, or in terms of conventional IVF, we wanted to learn from you about how you practice conventional IVF, because all of us have become ICSIs now, so we want to know more about IVF.
Correct. I have been using all these techniques for n number of years, and now I switched over almost to the swim up, because I have found that swim up is the one that is the simplest one, it is quickest one, and it gives you sufficient sperms for your purpose, depending upon the quality of the semen, of course. But the next alternative is the microfluidics. So I switch over between these two. Of course, I also go for the density gradient in certain cases. For example, if in case there are more debris is available, or more you found the leukocytes and pus cells are there, then definitely I will go for that one. But nowadays I am getting such samples very rarely, so I very rarely do the density gradients as such. But as I mentioned, in some cases, in case of PISA and TISA, I go for the single density gradient. So my first preferred way is swim up, and then the microfluidics, and in some cases the single gradient.
Okay, sir. So for all testicular sperms, do you recommend using activation, oocyte activation?
For all
testicular sperms, for ICSI, do you think
oocyte activation? No. Oocyte activation is not for that. See, basically, testicular sperms, most of the time, they had acquired. Yes, it is true that it is better if they pass through the epidermis. So they will acquire those particular coating of the proteins. Those proteins are necessary definitely to activate the oocyte. Activate oocyte in what sense? During the meiosis 2, before the expulsion of the second polar body, second polar body will be expelled after entry of the sperm into the ooplasm. Remember that. Generally, it is a misconception that the second polar body is observed. That means the fertilization has taken place. No. Second polar body, when extrudes at that time, it is because of that entry of the sperm. That sperm activates the oocyte, which is still at the diploid status with 2 chromatids. That one chromatid should be expelled out. That expels out in the form of second polar body. And then the sperm, after the entry of the sperm, that definitive egg becomes eligible for fertilization. And fertilization process starts after that. So it is necessary that the entering sperm should have that capability of activating the oocyte to undergo the second polar body excision. Now, this is generally observed that the TISA sperms, there is a mixed population. Some sperms they have it, and some sperms they don't have it. But generally, based upon the global research articles, it is observed that around 70% of the sperm, even from the TISA samples, they have the acquired proteins that activates the ooplasm to get that second polarity extruded. So there is no need as a routinely we should not go about it. But yes, if you have the experience of it, or previously it is already experienced that these sperms could not fertilize the oocyte, then definitely you should go for it. Or if you have more number of oocytes, then you can split the oocytes into two parts and then try few oocytes with this and few oocytes with that, so that you will not end up in a situation of no fertilization.
Thank you so much, sir, for patiently answering so many questions. Sindhu,
Thank
just
you.
check if there is any final question from the chat box. Otherwise, we can.
Since many were repeating, I've answered most of it from what sir has told. So
Somebody has asked one question. For conventional IVF, what is the standard concentration per oocyte and recommended drop?
It entirely depends upon the grade. I give my full attention to the grade of the motility, and of course the percent motility. Or percent motility, generally you get almost 100%. That is our aim. Whether you use swap technique or use the density gradient technique, the final supernatant should have almost 100% motile sperms. That is what is expected. But the grade is more important. If you have almost all with grade 4, then even a 5,000 sperms per oocyte is good enough, depending upon the droplet size. If you're doing it under the oil, you may go for even lesser than that. Because remember, you are putting the sperm in a medium droplet, and unlike the natural conception, the sperm doesn't have to go through the hurdles from the vagina till the end of the fallopian tube. They are there, they are just traveling, they are sitting around. So they don't have any obstructions between them and the oocyte. So they will immediately go and attach there. So the main task that is done by the other sperm is to quickly dissolve the cumulus part of it and allow the most eligible one to attach to the zona pellucida. You don't need so many samples for that, so many sperms for that. But yes, if there is a twitching motility, the grade of motility is only twitching and 1, then you may increase it to around 15 to 20,000 sperms, depending on if you're inseminating it into the droplet part of it. If you're using central well dish and then putting say around 1 ml or 1.5 ml of the medium, then obviously you have to go 10 times more than that, because you are giving more medium around the around that oocyte, and more distance should be traveled for the and sperm. Although there is some kind of chemotactic movement that takes place, the moment you put the sperm into the droplet of the egg, it will be attracted towards the oocyte automatically, but not necessarily that at one go it will go into that. If you watch the sperms put into the central well dish under the microscope properly, you will see that all of them are not going into the same direction. They are wandering around, so they may go exactly opposite direction than where the oocyte is. So these kinds of attraction and chemotactic movements are not the same for all the sperms, and this is the reason why we should increase the probability, so that in case if the sperm number is slightly less for the fertilization purpose, then they should not run short of the sperm, and around 10 times more you can go for it.
thank you, sir. We've taken almost 1 hour, 20 minutes of your time tonight. Thank you so
It's
much
my
for
pleasure.
sharing this time with us. And I think everybody has benefited a lot from this presentation and your points.
And
Good.
from beginning to end, we have a full house listening to you, and all your questions are, answered very, religiously. Thank you, all delegates. We wish you to, share your feedback, and also if you have more questions, please share it. We will, get answers from sir, or we will try to answer it. And thank you so much, sir. It was a privilege to have you as a mentor today for all of us.
Thank you.
Really, learned a lot from your lecture. We hope to do more from you in future.
Sure.
Thank
Sure.
you so much, sir. Thank you
Thank
so
you.
much.
All the a lot.
Thanks, Sindhuja. Thanks, entire team,
Bye and
for
all
us
the
to
best to
assist
all
us.
of you.
Thank you.
Thank you, sir. Thank you.
Thank you.
Thank you, ma'am.